which addresses transparency of the advice provided to federal agencies and the management of conflicts of interests by any of the members. So in the process of developing the committee's roster and preparing the nomination for non-federal members to join the committee, we've documented any significant financial interests and involvement in other organizations that could pose potential or perceived conflicts. Potential conflicts of interest among federal employees are managed within our agencies. So I've provided recusal and waiver lists documenting any such conflicts of interest to all the members who have such relevant financial interests. Members would be recused from discussions and actions affecting organizations with which they have conflicts. Today's meeting agenda does not involve any actions or discussion specific to the organizations or financial interests of our members, so we do not foresee the need for any such recusals. So the meeting is open to the public and is being recorded. We will post the recording and a summary of today's meeting in a list of the committee members participating on our website, which is mdcc.nih.gov. We have posted a notice for this meeting in the Federal Register and invited public comments. No one has requested the opportunity to address the committee, so we've not scheduled any public comments during this meeting. So I'd like to acknowledge the NIH staff that have helped prepare for this meeting, including Melissa Parisi, Tracy King, and Lisa Kaiser from NICHD, Wei-Ching Lee at NHLBI, Cariffi, who until recently was a health program specialist at NINDS. So Emily has been responsible for really a lot of the behind the scenes management of MDCC over the past 4 years, and I'm very appreciative for the excellent work that she's done. So Emily is now the program director for muscle diseases at NIAMS, and I look forward to her continued involvement with the MDCC in her new role. And thanks for helping out and still running WebEx for this last meeting. One of the important committee activities that Emily has led has been the collection and analysis of muscular dystrophy research support data from our member organizations. Thanks to all of you that have provided their data for 2019. Unfortunately, we don't yet have all of the data needed for that analysis, but we expect to complete that soon, and we'll put that on the fall meeting for presentation of that data and analysis. For this meeting, the committee members and NIH staff will normally be muted and can unmute yourselves to ask questions. Other attendees can use the chat function on the WebEx toolbar. It looks that thing that looks like a round bubble, like a cartoon character speaking. So click on that and just type to everyone the questions that you have, and I'll read those out for you. We're always looking for topics for future meetings. Our next meeting will be December 16th, and I've learned that all NIH council and other advisory type meetings for the rest of this calendar year will be held online only. So that will be, instead of an in-person meeting on December 16th, it'll be a WebEx like this one. But if you have suggestions for topics, please send me your ideas, and be happy to take any questions about this matter. Or if there are no questions, then I'll turn over the meeting to our chair, Dr. Bianca.
Okay.
Has Dev Gillipoli been able to join this list yet? So, okay, please go ahead, Dr. Bianca.
Thank you. Thank you, Glenn, and thank you to all the staff that you mentioned for all of the work that you've put in for organizing this particular session today. This is my first session as chair, and of course, we have a few complications, but we're now 3 months into doing everything virtually. So hopefully it'll go smoothly. And again, what I found is that the chat boxes have been actively used in most conferences and really allow a degree of engagement that we don't necessarily see in in-person meetings. So I've prepared a few slides, and so hopefully Emily can bring them up, and we'll get started. Excellent. So next slide, please. Next slide. Yeah, so I'd like to welcome everyone, and I'm just going to, everyone always is very interested in what's happening in terms of money and the budget. So we'll start with that, and then we'll talk about what's on everybody's minds, and it's affecting all of our lives, and that's COVID-19. And then I will be pleased to present a few highlights in terms of advances in muscular dystrophy research. Next slide, please. Next slide. So we fortunately do have a federal government that's currently funded through September 30th, 2020. The president's budget for 2021 was proposed a cut of 12% to NIH. That's what was proposed. We were able to do our testimony to the Subcommittee on Appropriations to the House in early March, but the Senate meeting was postponed or canceled, so we didn't get to do that. We understand that there are plans to mark up the 2021 budget, but everything is very much up in the air right now. However, we're very grateful to Congress for providing stimulus funds for COVID-19 through HR 748, otherwise known as the CARES Act. And NIH received $945.4 million to, quote, prevent, prepare for, or respond to coronavirus domestically or internationally. Specific targeted funding was given to NIAID, NHLBI, NIBIB, NCATS, NLM, and the OD. And we've had a number of activities in which the institutes not mentioned in that second bullet, as well as all of the institutes, have been writing proposals and trying to address needs of their particular populations to compete for some of the funding that went to the Office of the Director. Next slide. Next slide, please. Next slide. So I thought for those of you who aren't necessarily up to date with what's been going on at NIH in the past 3 months and how we're addressing COVID-19, I thought it would be helpful to just give a super broad overview, and we're happy to answer questions. I should mention that both Dr. Koroshetz and Dr. Carter are joining us, so they will also be able to answer questions specifically as they affect our 3 institutes. NIH has, particularly Dr. Collins, has been exceptional in leading a public-private partnership known as ACTIVE. ACTIVE is an acronym for Accelerating COVID-19 Therapeutic Interventions and Vaccines. And it's really quite remarkable. I don't know if you can see in the organizational chart on the right side, but you've got leadership from Johnson & Johnson, from Roche, from the FDA, from Pfizer, as well as NIH. And all of the industry partners have really set aside their proprietary interests to address this enormous public health crisis with the common goal of developing treatments and interventions as soon as possible. Next slide. So that's one major activity, and there are a number of working groups, again, that are devoted to vaccines and therapeutics. Another major effort is something called RADx, which is really focused on developing new and easier ways of testing for COVID, for scaling up the number of people who could be tested at a given time for COVID-19. And there are really multiple efforts. So what's shown here in this graphic is really what we are now calling RADx Tech, which is a Shark Tank-like rapid selection process. And the NIBIB received over 1,000 applications from people or companies that had ideas as to how we could develop new technologies for testing and how we can ramp up the testing process to be able to do tens or hundreds of thousands of tests in a week. Those RADx Tech tech proposals are going through a very rapid process where they're evaluated and a very limited number are then being offered to go to the next step. But in addition, we have multiple other RADx programs. So one is called RADx UP, which stands for Underserved Populations. And that is looking at how do we get testing out to people who either don't have access to health care, who are living in rural settings, who don't have health care as a basic right, people who are in prisons, children, all kinds of populations, to try to understand who has COVID, who may have it and be asymptomatic and therefore is at risk of spreading it to other people. So that's RADx UP. And then there's RADx RAD, which stands for Radical, and that is another group of proposals that are meant to bring new ideas in that may take a little bit longer. So this also provides some startup funds. And then the last one is called RADx ACT, meaning that there's activation energy needed to bring these proposals for testing. They may need a little bit longer. They are that much more innovative or need additional funds to really be able to scale up the testing technology. So those are the two major efforts: therapeutics, vaccines, interventions, and diagnosis and testing. Next slide. Since a number of the conditions that we're concerned with in this particular committee, and certainly at NICHD, we're very concerned about manifestations of COVID in children. And until about mid-May, we thought, well, children are really not so seriously affected. We knew that there were many children who had mild respiratory illness, but we certainly hadn't heard about children dying until mid-May. And then reports began to come in from Europe and also from New York City that a small subset of children were developing a mysterious multi-system inflammatory syndrome that seemed to occur about 3 to 4 weeks after having had either a mild COVID-19 positive infection or kids who tested positive but who weren't sick at all. But then 3 to 4 weeks later, there seems to be this robust immune reaction that in some cases resembled Kawasaki's disease, and in other cases was something completely different that people really had never encountered before. And we are just in the beginning stages of a multi-institute partnership that predominantly involves NICHD, NHLBI, and NIAID, along with the CDC, the Office of the Assistant Secretary for Health, and others, to begin to address who is at risk to develop this condition, what are the best ways to treat this condition, and importantly, what are the long-term effects of this condition. Next slide. So let's just quickly talk about some highlights of muscular dystrophy research, and we've selected highlights from each of the institutes. So we have one here from NINDS, which was published in Scientific Reports about identification of utrophin upregulators as a potential therapeutic. And upregulating utrophin can functionally compensate for the lack of dystrophin caused by DMD mutations. And this study conducted a high throughput screen for small molecules to increase endogenous utrophin expression. The investigators identified 27 potential hits and then ranked them and validated the top 10 of them. And the top scoring hit was trichostatin A, which showed functional improvement in the mouse model of DMD, the MDX model. So TSA and other small molecules represent potential starting points for drug discovery. Next slide. In a study that was supported by NIAMS, NHLBI, and NICHD, CRISPR-Cas9 was used to make single cuts to correct genetic mutations in animal models requiring high doses of adeno-associated virus, or AAV, and that limits clinical application. So these researchers used a modified delivery system with self-complementary AAV, known as scAAV, which has previously been used in gene replacement trials for spinal muscular atrophy and limb-girdle muscular dystrophy. And in a mouse model of DMD, the dose required for efficient genome editing was much lower. The mice showed restoration of dystrophin expression and improved muscle contractility. So another potential tool that could be translated to DMD therapy. Next slide. And speaking of mouse models, there are a couple of new mouse models for facioscapular humeral muscular dystrophy. And these are mice that show expression of DUX4. This work was supported by NINDS. Next slide. And so interestingly, again, looking at DUX4, in this particular study supported by Ph.D. and NINDS, again, CRISPR-Cas9 was used to knock out muscle cells expressing the DUX4 protein. And they removed genes one at a time using CRISPR-Cas9 to find the ones in which cell survival improved without a particular gene. And they discovered that the cell's response to hypoxia was driving cell death in affected cells. So hypoxia signaling inhibitors enhance the cell's ability to remove the DUX4 protein and reduce signs and biomarkers associated with FSHD disease progression in actual patient muscle cell lines. And they improved structural and functional properties in 2 zebrafish models of FSHD. So this, again, if you could interrupt this typical process for FSHD, this could have therapeutic merit. Next slide. This is published in Science Translational Medicine. And then a work from intramural program at NINDS, there were mutations identified for a new phenotype of early onset muscular dystrophy. So this is interesting because it has not only muscular dystrophy, but it's associated with hearing loss and primary ovarian insufficiency. So that particular combination is quite interesting. This particular study that was published in the Annals of Neurology performed whole exome sequencing and identified variants in the gene GGPS1, which is, this particular gene is involved in making geranylated proteins, including small GTPases. And there's a potential novel pathway for this phenotype. So we look forward to learning more about this new condition. Next slide. So I think those are in my very brief time on exactly at 1:25. So I think Glenn hopefully will be happy with my ability to keep to time in my first role as chairman. And so now it's my pleasure to turn the meeting over. First of all, for each of the meetings, as you know, we first invite brief reports summarizing meetings and conferences of interest to the entire group. Of course, we're in the middle of a pandemic, so most meetings have either been canceled, postponed, or are occurring only online. So today we're going to hear from Ryan Fisher from Parent Project Muscular Dystrophy. We know he's on the line, and also Deborah Miller, who we saw on the line, who's from Cure Duchenne. So they're going to tell us about the meetings held prior to the shutdown. So first, Ryan, could you please tell us what you have to tell us?
Thank you, Dr. Bianchi and Dr. Nuckolls and members of the committee. Can you hear me okay? Am I coming through all right? Okay. Hopefully I can also keep to my time and not go over. So while the slides are being pulled up, I'm going to present on a meeting that we held in early March. We actually got right under the wire before things started to change in terms of shutdowns and stay-at-home orders. So We were able to do this meeting in Washington in person. We normally hold a meeting in conjunction with our annual fly-in, and it's usually multi-stakeholder and involves bringing together federal partners, members of industry, and of course, members of the patient community on a given topic. And this year marked an important anniversary in our view. It had been 20 years since the first ever NIH workshop on Duchenne muscular dystrophy that was organized following a request by PPMD in 2000. And we wanted to use this opportunity to mark this occasion and sort of reflect on where we've come from that first initial NIH-sponsored meeting. So you could do the next slide. The meeting was entitled The Duchenne Community and Federal Research Agencies, Yesterday, Today, and Tomorrow. We worked with many of you who are on the line today to put this together, and we're grateful for your contributions and your time. Here's a look at the speakers on your screen of those who were part of the speaker and panel presentations. You know, and then some of the objectives. We felt it was important to convene the community in a discussion with our agency partners to really reflect on what we've done collectively to date, including the research that's been funded both publicly and privately, discuss what's been learned, and importantly, consider priorities moving forward and how we essentially could continue the momentum that was really started 2 decades ago. You can go to the next slide. We also took time to recognize the contributions of Dr. Stephen Katz, who I know many of you knew and respect, longtime director of NINDS, and who we sadly lost in 2018. Dr. Katz was former chair, actually, of the NDCC and was really critical to the implementation of the MD Care Act. But importantly, he was a friend to so many in our community and other communities that he served and made a huge impact in advancing the understanding and treating of many diseases. So next slide. We, because of those contributions, we actually named a new award after Dr. Katz, and awarded the very first one to the first speaker at our meeting, Dr. Stephen Groft. Many of you know Dr. Groff has made a huge impact on rare disease, and this award was well deserved, given not only his efforts in broader rare disease, but he happened to be the one to help lead that first Duchenne meeting 20 years ago at NIH. So we certainly felt like we were coming full circle with acknowledgement of Dr. Groff. So it was a nice support prize to award him with this award. So next slide. And Dr. Groff sort of started the day with a look back. And here on your screen, you actually see there's a link there, and there's a way to access this very first workshop. We actually were able to find the report. It's a really remarkable thing to read when you look at the specific areas outlined and identified from that meeting, really some of the building blocks to enable the research necessary for drug development, things like genotyping and phenotyping, and how to advance gene therapy, cell-based therapies, new molecular entities, like high throughput screening of new compounds, assessing steroids. The list goes on and on, you know, MRI, non-invasive techniques of measuring progression. So lots of areas identified during this 3-day workshop in 2000. You can go to the next slide. And just really the priorities that were identified, obviously, you know, addressing those, one of the things that came out of that was the legislation, the MD Care Act, and I don't think I had to spend much time on this, but the MD Care Act and subsequent amendments really are the reason why we are all here today as well, where NDCC was authorized in the bill. You can go to the next slide. And just comparing to where we were prior to 2000 and where we are now, just by some different metrics, Dr. Groff reflected on things like orphan product designations, publications, and even drug approvals, really showing progress of increased investment towards Duchenne research and therapy development. The next slide. And here we are today with over 30 clinical trials and a large number of preclinical studies happening, not on your screen, but none of this would have been possible without the collective work of the federal agencies and patient advocacy groups, involvement in basic and translational research, driving forward these early good ideas into translating them into later stage clinical studies. It's really been an extremely collaborative effort on so many levels by many of you on the line today and the institutes and agencies you represent. So this meeting really allowed us to reflect on some of that and the acknowledgement of that work, as well as the realities of there's still a lot of work to be done. So next slide. We did talk a bit about resources that were available for patient groups. Many of you might be aware of this toolkit that was developed in collaboration with NCATS, and PPMD was one of the co-chairs of the committee. The purpose really is to assist patient groups in creating and advancing their research agendas through patient-focused means. And then next slide. So we spent the bulk of the meeting discussing the current research happening out of some of the key institutes and then the Department of Defense. Excuse me. And Center for Disease Control, Natalie was invited, but at the time with all that was happening, obviously they were unable to come in person. So we are planning a follow-on webinar with CDC, and we'll let you know when that's happening. But you know, here's just a look at some of what was discussed. You know, both the NIH and the Department of Defense, I think everyone knows, has invested so many resources, time, and energy across, you know, really all aspects of drug development in Duchenne and in other diseases, everything from understanding the natural history to developing biomarkers, advancing therapeutic targets and new mechanisms of action for drugs, and the recent advancements in gene therapy and gene editing really all can be traced back to and connected with the ongoing investment at the federal level. We had some great presentations from the institutes and from the DoD highlighting some of the ongoing work and reflecting on the past 2 decades. Dr. Nuckolls gave really the first talk to provide an overview to the audience on the Muscular Dystrophy Coordinating Committee and the action plan. He also touched on work out of the Wellstone Centers, highlighting some of the projects out of NINDS, including Vamorolone and AAV gene therapy and exon skipping. Dr. Cheever spoke on projects out of NINDS, including AAV gene therapy and gene editing, as well as the advancements, really incredible advancements made with MRI non-invasive ways of measuring disease progression and therapy impact. Dr. Parisi spoke about where we were in newborn screening and the work out of NHCHD. And then finally, Dr. Kiley gave a great overview of the work out of NHLBI, highlighting studies with the Plerinone and some of the recent work with Eric Olson on CRISPR. And then Dr. McGuire finished the day from DoD to present on the research out of the CDMRP. What is exciting there is a recent increase in budget, and so the opportunity to fund more great projects. And Dr. McGuire presented on past and present projects out of DoD. Next slide. So we closed the meeting with just some discussion on where we go from here. We had a panel moderated by Annie Kennedy, whom you all know, and I think the consistent theme across all speakers was just to continue building on what's been accomplished to date over these past 2 decades. We've made major progress in drug development, working together, forming these collaborations across federal partners, patient advocacy, clinical care, industry. I think everyone in the room shared that desire to continue those efforts and partnerships, and to include pursuing all strategies to treat Duchenne, from foundation therapies aimed at DNA and RNA, all the way to the downstream targets on inflammation and fibrosis. So knowing full well that we need combinations at the end of the day to treat Duchenne muscular dystrophy. And in parallel, we need to continue our work on understanding natural history, which really has matured over the past 2 decades, where Duchenne is much better characterized than it was 2 decades ago. So continue to further that characterization was critical, including unsiloing data that exists that could aid in that whenever possible, and continue to advance measuring disease progression in different ways like MRI, including muscle and cardiac MRI. So we touched on some of the use of real-world evidence and more representation of patient perspectives, as well as improving patient-reported outcome measures, which we're going to talk about today. And in closing, I just think it was a great opportunity to reflect on where we've come from and discuss where we need to go next. So once again, thank you to so many of you who participated in that meeting. Feels like a lifetime ago, even though it was only in March. But we really did appreciate the conversation with the community. So with that, I will hand it over, I think, unless there's any questions, to the next speaker.
Yeah, so thank you very much, Ryan. I don't know if there are any questions for Ryan. I do see that Daniel Perez has written something in the chat box, which I'm not sure if that was meant for me. I was so focused on the time, quite honestly, I forgot to give everybody a chance to ask any questions. So feel free to use the chat box, which again, I think people have been using in most of the NICHD workshops in a very productive way. Does anybody have a specific question for Ryan about this forum and the incredible 2 decades of progress in research? If not, I just do want to acknowledge Daniel's question, which is in the chat box, and it says, many individuals with muscular dystrophy die from respiratory failure, such as acute respiratory failure, metabolic disturbance, and acidosis and clotting. Can you comment if increased demand in this area vis-a-vis COVID-19 is improving care? Is there a better handle on carbon dioxide retention, et cetera, and opportunities, I think, for grant funding in this area? So I don't know whether, Dr. Kiley, do you want to answer this from NHLBI's perspective? I could say from NICHD's perspective, right now we're really focused on children and pregnant women, not specifically subsets of children with particular conditions. Does anybody want to answer either via the chat box or put on their microphone?
So Diana, this is Jim Kiley. So certainly we've seen a lot of increase in quality of care and the approach to patients in the intensive care units, as well as throughout the hospitals and as outpatients in respect to best management of acute respiratory diseases. We know that a lot of the COVID associated with comorbid conditions, asthma, COPD, other conditions underlying, but also heart disease, seem to accelerate, precipitate worse outcomes. So we've learned a lot. So in part, the beginning of that question is that I think care is probably definitely getting improved in terms of how we're managing the patients. It's unfortunate that we've had to go through this to see that happen, but I think we are getting a bit of a spin-off from that. So yeah, for sure, across the whole spectrum, not only in NHLBI, where we've put out a lot of announcements for notices of special interest to stimulate research in this area, particularly in the heart, lung, and blood areas where a lot of action occurs in the lung and in the endothelial cells in the blood and the coagulation system, which is central to NHLBI. But I would have to say it cuts across all of NIH. Dr. Bianchi mentioned about MISC. I mean, that's clearly growing, and we've seen enormous amounts of efforts coming in from the scientific community just right across the board to try to understand this disease better, both from a very fundamental basis all the way up to how we can treat it better, how we can come up with vaccines. You've already heard about how we can do better point of care diagnostics in all kinds of communities. So I just say across the board, NIH has really stepped up its game, and we are collaborating extensively, probably in an unprecedented way over the past few months. And then I'll just wrap it up by just saying that the response from the community has been phenomenal. I think, again, Diana mentioned how industry has come to the table. Every single investigator across the country, probably the world, that has any relationships with NIH has come forward with either ideas or willingness to bring patients, samples, whatever, to the table and try to help accelerate our understanding of this problem as well as ways to best treat it. So I think we are putting out a lot of announcements. There's a lot of things going on. I would say check regularly the NIH website as well as the Institute websites, because there's just a tremendous amount of opportunities for people to help address this growing and a huge public health problem.
Thank you, Jim, for that answer. You also reminded me to share that there is a portal that NIH has for unsolicited ideas, either coming from individuals or small companies. And there's a formal mechanism where if you have a great idea that you think will improve either treatment or diagnostics, you can share your idea and then it will be reviewed by each of the institutes and it potentially can go forward. So it really is a community approach to tackling this incredible public health crisis. So thank you, Dan, for your question and Jim for the response. So now I'd like to go to, yes.
If I may, this is Kristen Stevenson with the Muscular Dystrophy Association. I just wanted to mention from a clinical management standpoint, particularly around respiratory care and the interplay with COVID-19, there is a resource on the MDA website specifically around clinical care guidelines for individuals living with muscular dystrophy that have COVID-19. And there's also a Facebook Live event this Friday that will be specifically addressing respiratory needs and pulmonary support in this time. So just wanted to flag that for the group.
Excellent. Thank you very much, Kristen. So now we'd like to turn to Deborah Miller from Cure Duchenne. She's going to give us an update on the 2019 Futures National Conference and the launch of the Cure Duchenne Biobank. Deborah?
Great. Hi, thank you very much. Can you hear me okay? Great. Okay, and thanks Ryan for such a wonderful presentation and your work on behalf of the Duchenne community. And I really want to say thank you to the MDCC for inviting me to present the Futures National Conference and the launch of our biobank. Next slide, please. So the vision of Cure Duchenne is to improve the lives of everyone affected by Duchenne muscular dystrophy. And we're committed to improving the lives of everyone affected by this disease by accelerating research to find the cure, redefine care, and empower the Duchenne community. We have different programs, and you'll see the logos listed here on the right side of the slide. I'm going to cover most of these, but a couple that I probably won't be covering today would be Cure Duchenne Ventures, which is our research and investment arm. And we're proud to say that over the past many years, a lot of our early research has ended up progressing into what are now the clinical trials and approved drugs for Duchenne. And then also the Duchenne Exchange, which is a free collaborative online platform for patients and families and advocacy organizations to connect and share knowledge. And so, but I'll go ahead and talk about the other programs here today. Next slide, please. So we started off about this journey started 18 years ago for me when our son was diagnosed with Duchenne muscular dystrophy. And back then there were very few healthcare providers in our area that were focused on Duchenne. And we were lucky to find a physical therapist even before the actual diagnosis, Jennifer Wallace. And Jennifer had recently received her degree, and we were fortunate that she took an interest in our son and in Duchenne overall. And as word of Jennifer's work spread, we had requests from around the country for her services. And wanting to replicate her expertise, we created Cure Duchenne Cares, which is a national series of workshops for families and healthcare professionals. And with our scientists and physical therapists, we travel across the country to communities large and small where there's a need for specialized training. And starting off many years ago, the Cure Duchenne Cares program in 2019 actually grew to 27 events in 22 states and 27 cities. And through the Cure Duchenne Certified Physical Therapist Continuing Education Series, we held in 2019, we held 6 professional courses. These are continuing education, trained 112 therapists, and 22 of the therapists went on to receive the advanced certification of the Cure Duchenne Cares program. And every year we hear from parents that have had bad experiences with therapists that didn't understand the nuances of Duchenne. And we're really proud to have prioritized the importance of physical therapy in Duchenne and increase the number of qualified PTs. Next slide, please. So after 8 years of regional and community workshops across the country, we realized a significant benefit of these events was the social aspects. And so we realized the need for positivity and practical solutions for the families. We launched an annual national meeting in Boston in 2018. And in 2019, the Futures Conference grew by leaps and bounds with 585 attendees from 37 states and 14 countries. And our panels our panel format allowed for more speakers at the conference and allowed real life settings to occur, meaning patients and physicians being on the same panel, mental health care providers with parents on the same panel. It was all very interactive. And so we also were able to entertain the kids and provide a lot of socialization for the kids through our gaming lounge. We had over 100 participants on that. We provided childcare for over 100 kids with 45 professional nannies. And we were able to not only provide free registration, but also provide a lot of travel assistance. And we also had an opportunity to work with some of the sponsors to conduct focus groups with patients and caregivers. Next slide, please. So we have been focused on our conferences. The Futures Conference is the 5 F's: family, fitness, friends, fun, and function. And so we really try and present a very comprehensive environment for families, for physicians, and all of the healthcare experts to interact. And so we had KOLs highlighting the standards of care, all the way from pediatricians to neurologists to cardiologists. We had a geneticist and genetic counselors addressing the genetic needs of the whole family. We had psychologists and mental health care counselors discussing the stages of grief and coping strategies, special needs attorney, insurance specialists discussing daily supports and needs, our soccer coach and adaptive ski developers that were sharing tools to access the environment and enjoy life, and young adults living with Duchenne, and school-based therapists discussing communication and relationship building. We also really focused a lot of the research into 2 significant panels. One was an exon skipping panel with all of the industry partners, and another one was a gene therapy symposium dinner where we had all of the industry sponsors and some other researchers to discuss the present and the future of gene therapy. Next slide, please. So it was really important for us to understand, how to judge ourselves and our success on this event. Our goal was really to listen to the families and to understand their needs. So we conducted a pre-event and post-event survey. And as you can see, we actually believe that we moved a lot of education forward in this 2.5-day event. From understanding clinical trials, we provided a lot of information that helped increase their knowledge, as well as how to access the clinical trials. Only 63% of the attendees were aware of home stretching techniques prior. After the event, 93% were aware. Emergency care considerations jumped from 53% up to 89% post-event. And financial assistance went all the way from 31% up to 77% after this event. And so we're always striving to ensure that the attendees have a meaningful experience. And so we also found that 73% enjoyed the gene therapy panel and wanted more information. 82% said the content was relevant and they felt they were given time to ask the right questions. 86% enjoyed the panel discussion. And one thing that was really important about this conference was the social aspect of it. It was a very friendly and fun environment also. Cure Duchenne, prides itself on its events of really focusing on the positive and solutions. 91% valued the socialization with families, speakers, sponsors, and the Cure Duchenne staff. We also pioneered the expert meetup, which was a really valuable tool where the families could pre-register for appointments with individual physicians or scientists or psychologists, and that was very, very popular and useful. When it came to the focus groups, 100% felt it was neutral or positive, and most of them said it was tough, but it was good and necessary. And so, I think that it may not be the most pleasant thing for some of the families to participate in, but I think they really understand the value of it. So, our work is just to ensure that there is as much knowledge, which will lead to empowerment of the families. And we want them to be able to access all sorts of resources like vendors, wheelchair vendors, services, and products that improve their lives. And we were really excited to launch the Cure Duchenne Biobank. So if you go to the next slide. So the Cure Duchenne Biobank came about as we're looking at the future of more personalized medicines and also looking for new targets within diseases. We received multiple requests for biosamples from researchers, and it seemed that a lot of people are having trouble accessing these. So in order to fill that gap, Cure Duchenne created the Cure Duchenne Biobank, and this is an independent biobank. It's not funded by pharma, and it's available for all qualified Duchenne researchers. And at this time, we're collecting blood and skin samples. We partnered with the University of California, Irvine, with Dr. Tassim Mozaffar and Dr. Armando Vialto, and RUCDR is hosting the biobank. And we're really committed to reaching out to underserved communities and populations. We were collecting these samples at our Futures Conference, and we were actually very pleasantly surprised by the response we received from the families that wanted to participate, and they really wanted to have their child's samples tested and part of the research. And so we're taking samples of siblings also that are unaffected. And so we were, again, very, very excited about the response we had. We were taking additional samples at our Cure Duchenne Cares events around the country. Unfortunately, COVID hit, and so that's been on pause a little bit, but we're looking forward to starting up on that again. Next slide, please. So we are really, really excited about the future that the Duchenne community has, and we also have created a COVID resource. It's called HUG, H-U-G, and it's on our website, and it covers everything from emergency care to respiratory to state and community resources. And so as the world has pivoted to virtual, so has Cure Duchenne. We have COVID-19 response webinars. We have coffee talks to bring communities together and try and still keep a local community flavor so that families can meet each other in their own areas. And we're also hosting a webinar series called Embrace This Moment, which is a 3-part webinar series that will run through June through July. We're really breaking it up into components of the journey. So newly diagnosed families, mid-stage, and then families with older patients or older boys. So we're really proud of our COVID response, and our goal is really to continue lifting up the patients. And we're just very grateful to all the experts and everybody that's participated in all of these, and really dedicated to continuing to educate the families throughout the summer and hopefully in person very soon. That next slide.
Thank you, Deborah. Are there questions? Yeah. So we'll be very interested, maybe at our next meeting, you could report back to us. Certainly what we were hearing from the workshop that we held with the Down syndrome community was that some of these virtually only opportunities, for example, the opportunity to participate in research online or to participate in conferences online, allowed a level of accessibility that made it easier for families. So I personally would be very interested in your perception of how things are different. You know, there may be some positive things that come out of this in that you may be able to reach families that for whatever reason have had difficulty traveling to the locations where your meetings have occurred. I don't know if you have any thoughts on that.
Yeah, it's, you know, I spend most of my day on Zoom these days, and, you know, I'm, you know, back in the office today with a very limited staff here, and it's nice to be back, but at the same time, we have reached so many people. And it's been so accessible, and it's recorded, so they can, you know, if they can't make the particular time slot, they can tune in. There is the lack of the social interaction. I mean, I think there's, you know, with Zoom and seeing everybody's face, it's helpful, but it's not quite the same. So I will definitely report back to you. And I think moving forward, a combination of in-person and Zoom meetings is what we'll probably be expecting.
Great. Well, thank you very much for your presentation. So next, we've invited Michelle Campbell from the Center for Drug Evaluation. Yes, sorry.
Hi, this is Glenn. I just want to give an opportunity for a few members who joined and we missed them in the introductions to introduce themselves. So I know we have on the line now Dev Gillapalli.
Hello, this is Dev Gillapalli, Medical Officer, Food and Drug Administration.
Thank you, Dev.
Welcome.
And Cheryl Williams.
Good afternoon, everyone. This is Cheryl Williams. I am from the Social Security Administration, the Director of the Office of Medical Policy.
And I think we missed Mary Elena McGuire earlier.
As Glenn said, Maria Elena McGuire. I'm with the DoD's Duchenne's Muscular Dystrophy Research Program.
Welcome.
I know we also have Teresa San Agustin. Now there's a delay in audio to her text interpreter, so she may not be able to get on and introduce herself, but she's a longtime member of the committee, very active participant in our committee and representative from the Administration for Community Living. Are there any other members who we missed in the introductions? Okay, thank you, Dr. Bianchi. Back to you.
Okay, thank you, Glenn. So next, we've invited Michelle Campbell from the Center for Drug Evaluation and Research at the FDA to participate in our session later in the agenda on patient-reported outcome measures. A few days ago, the FDA announced a request for information and comments regarding practical steps and successful approaches to establish rare disease clinical trials, to establish a rare disease clinical trials network, which we thought would be of significant interest to this group. Thankfully, Michelle was also willing to tell us about this planning process. So Michelle, we're looking forward to hearing from you.
Well, thank you, Dr. Bianchi, and thank the MDCC for inviting me today. So as been mentioned, my name is Michelle Campbell, and I lead stakeholder engagement and clinical outcomes for the Office of Neuroscience in the Office of New Drugs in CDER. And so if you haven't been following the new drug reorganization, we now have new offices within the Office of New Drugs, and the Office of Neuroscience now captures what was our Division of Neurology Products, which has been split into two, our Division of Psychiatry and Division of Addiction, Analgesia, and Pain Medicine, as well as a much larger division of Pharm Tox. So we are part of the new Office of Neuroscience there. And so this is a great opportunity and time to speak about another project that I am working closely on, is something called the Rare Disease Cures Accelerator, and I'm really happy to be able to present to this group today about the work that we've been doing for the last year, and also what this new RFI that came out just last week from the agency on a clinical trials network. Next slide, please. So I am speaking to experts as well on knowing what our challenges are in rare disease drug development. And so we know that oftentimes we all are struggling with the same challenges. And for us, we know it's typically is rooted in the lack of understanding of the natural course of disease with our small populations. It's often hard for us to look at randomized clinical trials, and we know that we need a better solution. Next slide, please. So what you see on your screen right now is just some notes that actually Dr. Teresa Mullen, who's our Associate Director for Strategic Initiatives, jotted down one day when Dr. Woodcock was speaking. But what are some of the challenges when we think about drug development and really the process of going from, you know, preclinical to actual clinical development when we're trying to think about what is a molecule that may work? And so it first starts with how do we characterize the disease? What do we know about the disease? What's the natural history? What's the cause of the disease? How do we get our patient perspectives in the disease? What's important to patients? What is currently even available in terms of treatment options? And then if we do have something that we can finally progress into clinical development, you know, our challenges include how do we design a study protocol that we can make sure that we are using the study population that's available because we know we have limited numbers. What is the plan for data collection? Do we have enough safety data to be done so we can know that we'll be able to safely test this into humans? And so these are some of the challenges that we face, and these are just some of the limited challenges when it comes to the drug development arena. So next slide, please. So Congress appropriated to the agency in fiscal year 2019 funds specifically for the investment in innovation for rare diseases. And so CDER received the funds, and we have developed something known as the Rare Disease Cures Accelerator to work on this innovation. Next slide. So why do we need the Rare Disease Cures Accelerator, which we have abbreviated to RDCA? Well, we know we need to adopt a cooperative research approach to accelerate and move from the bench to the bedside. We really need that aspect of the translational research to be working together. We need, and we hope that this Rare Disease Cures Accelerator would provide that infrastructure for a cooperative scientific approach to really aim for clinical trial readiness in our rare diseases. We've actually already been having conversations with Ann Paris and NCATS and Tina Irv, who I also know is on the call today, with some of these conversations with clinical trial readiness and the RDCRN. And so this is one aspect we hope to be focusing on. There are three key components that we really see that this Rare Disease Cures Accelerator will incubate and really bring to fruition. The first is we would like to have a centralized and standardized infrastructure to support and accelerate rare disease characterization. As Ryan said earlier, in Duchenne's, we really have a good sense of natural history now, and that took many years to finally get that natural history, and we're still learning from it. But that is just one example where we do have the natural history, and often we do not have sufficient natural history in a lot of our other diseases. And so we hope to be able to kind of accelerate, if possible, our understanding of these diseases and being able to characterize them. The second key component is standard core sets of clinical outcome assessments. And we really hope for this because we know we have to measure the impacts that matter most to patients. And we would like to be able to look at core clinical outcome assessments that may be applicable to more than one rare disease. There's work already happening in this area through another set of funding opportunity that happened actually right before this appropriation came through last year. That is also being funded out of the Teresa Mullen's Office of Strategic Initiatives. And so some of this work is happening, and in the rare disease space, it's going to be some information on pain and physical function for right now. And finally, the third bullet is really, can we come together and put a global rare disease clinical trial network? Because for us, these trials are global. Because with that patient population, we need to make sure we can get every patient possible. And this is where this request for information came out last week. I'll get to that a little bit more in a second. But these are the three components that we really see as the cornerstones of this Rare Disease Cure Accelerator initiative that we've undertaken. Next slide. So we see that the data characterization is really going to be the foundational piece that we need to make sure we establish early and focus in on. Because we know that in order to have effective, comprehensive characterization of a natural history for a rare disease, we need to be starting this early. And so this is why we are focusing, for the last year, we've been focusing on very much on the rare disease characterization and this data and analytics platform. We need the characterization to be conducted rigorously with attention to establish data quality standards. And this will help us be able to design the best appropriate clinical trial designs and assist in for regulatory review when things finally do make it into the agency for us to review. We hope that we can standardize rare disease natural history study data platforms to make this a sustainable project. We hope that this platform would provide a disease-neutral background data framework so that we can be able to query among disease groups. We can add uniquenesses for particular diseases, but all have the same foundation and fundamental framework to start with. We know that disease-specific needs will be layered on, but that we're not just only focusing on one disease area, but hopefully we can start looking into the broader sense of disease work. We really hope that the primary objective of this rare disease accelerator will be to establish a data management and data repository system that can be used to house data from existing and planned rare disease clinical trials, and will contribute from many different stakeholders and organizations. We hope that this platform will leverage existing scientific expertise in data management and rare disease knowledge with partnered scientific organizations, and that from this platform we'll be able to build clinical trial readiness, identify gaps where we need to continue to do additional work, specifically occurring in the pre-competitive space to foster innovation for patients and, with rare diseases. So next slide, please. So this first foundational part is known as the Rare Disease Cures Accelerator and Data and Analytics Platform. And it truly is meant to be a neutral, independent data collection hub. That we want to promote data sharing with this because this is critically important, and to be able to share data across rare diseases in order to accelerate the understanding of disease progression and to optimize clinical trial design. Next slide, please. So we have funded the Critical Path Institute, which is based in Tucson, Arizona. We have been working with them collaboratively for over the last 10 years at the agency on many various projects, including prior work in data sharing in the neuroscience space with some of their disease-specific consortiums, with the Duchenne Consortium in particular being a part of this, in data sharing. And also partnering with the Critical Path Institute, it's a national organization for rare disorders, to help with the communication and the patient group aspect of this initiative. The schematic you see on your screen is really this vision of this platform, where on the left-hand side, we would be able to take in all different types of data sources from various stakeholders. So our clinical trial data, our observational data, our clinical studies data, patient registry data, all different various sources. It would come in and it would be curated and then ultimately standardized into an aggregated database and then be available for various different users, including industry sponsors of clinical drug trial programs, academics who are doing work. And so we want to be able to have this platform so we can get actionable changes in rare disease drug development. Next slide, please. So this project, as I mentioned, we had a launch meeting last September, and we plan to have our second meeting this coming October, and I can send Glenn information about that once we have additional details. We're trying to final determination if this will actually be in person or web-based. But I can definitely send out information now where you can access last year's kickoff meeting on this initiative. But what you see is while this past year, the Critical Path Institute, who is really developing this platform aspect, has really come up on the right-hand side. You'll see the red arrow pointing down. The different levels of interface that depending on the user type would be able to see. So there may be something very simple, such as the top one, that would help maybe provide simple descriptive information about diseases pulled from various data sources to help maybe understanding of prevalence or what kind of information is being collected in various studies. The next few levels you see after that is where we hope to be able to get into more analytics. So can we do some disease modeling of progression? Are we seeing when we've pulled together various data sets, are we missing something? Are we not capturing something that may be useful in helping us understand disease progression? The Critical Path Institute has had some past experience in this, like as I mentioned, in some other diseases, including some rare diseases. And so this has been very informative in the drug development space, and we really do hope that this will continue, and we hope this initiative as well will really help all of our stakeholders consider data sharing their data that they have into this platform to be able to be pooled with other various data sets. Next slide. So specifically for the RDCA, our long-term goal for impact is this centralized and standardized infrastructure to support and accelerate rare disease characterization. We hope to also standardize data that can be extracted in our CDISC format, which is a format that is required for regulatory submissions. And we know that data standards is a really important topic with a lot of different groups. And I know that we've had conversations with NCAS just a couple weeks ago about data standards. And so this is a big initiative. We hope that is a great a secondary win that we can get where we can be able to standardize our data collection in a more formatted way that can be used for many different users in the end. We hope that we can be able to optimize our clinical trial protocols for therapies. It would be great to really be able to help us look at what maybe be the best sample size available because of these simulation tools and know what exactly do we need. Because we have so few patients in our rare diseases, we want to make sure we maximize what our trials the best we can, and we really hope that this analytics aspect will help with that. And finally, I'd like to just touch on the final bullet when I talk about this project, because here we talk about historical match controls and contemporary controls to enrich placebo arms and reduce sample size. And we know that external controls has always been a very important topic in our rare disease communities. But we also know that we need to have sufficient natural history understanding and historical controls to be able to actually enact something like this. And it's actually a lot harder to do than one may think. I think that this would be an ultimate, absolutely great goal in the end if we could actually get that in our diseases where we have so much available rich data, that data sets are coming in from various sources that can really help us be able to do something like that. And I know this is probably a very important thing to our rare disease communities if this could actually happen. We really do hope this can happen. Next slide, please. So I just want to end quickly about the request for information on our rare disease clinical trial network. As mentioned, it was announced last week in the Federal Register that we are requesting information on rare disease clinical trial networks. So At FDA, we don't necessarily have much experience in this area, and so we want to learn from our other stakeholders who have had experience and what are those thoughts about this. So we want to know what is currently being done. What needs to be done to address clinical trial readiness to be able to launch a network? What are the important practical steps to be able to implement and sustain such a global trial network? And what are the considerations for such networks for the various range of sizes of our 7,000 known rare diseases? So we do ask for people who have comments to please submit it to the docket to help us. And docket comments are due at the end of July, on July 31st by 11:59 p.m. We are just doing the early stages of trying to see what this would look like. And so really, anyone's prior knowledge, experience in this, we would greatly appreciate you submitting a comment on the docket so we can take a look at it. We know that there's a whole range of stakeholders that would be important to this. Our NIH colleagues is one. From your experience in the clinical trial readiness grants that you fund and lessons you have learned. We know that our industry colleagues is another one, our patient groups. What are those considerations? So this is really an opportunity to bring your voice to the table as we start to think about this early, about what should we really take into consideration as we try to map out what a clinical trial network will look like for rare disease. Next slide. I think that should be my final slide. So I thank you for listening to me about the Rare Disease Cures Accelerator, and I will take questions, or you can always reach out to me later on if you think of something as you mull over what you've just heard.
Thank you very much, Michelle. We look forward to hearing more about this as the plans progress. And I think in the interest of time, either use the chat box or contact Michelle directly. And we're going to hear from Michelle later on in the program as well. So next, I'd like to introduce Dr. Tina Irv, who we know well at NICHD. She's a former NICHDer, but she moved to NCATS, where she now manages the NIH Rare Diseases Clinical Research Network. And she's going to tell us about a study of COVID-19 in rare disease patients that is being conducted by the network's Data Coordinating Center. Tina, thank you.
Dr. Bianchi, I'm happy to be working with my old NICHD colleagues again. So today I'm here to talk to you a little bit about a project that we're doing through the Rare Disease Clinical Research Network. Next slide, please. So for those of you who might not be familiar, the Rare Disease Clinical Research Network goes back about 15 years. Right now, we have 20 consortia. We have a data management and coordinating center. With these groups, we have about 140 patient advocacy groups that are significantly involved in all of the activities that we have going on. We are funded by over 10 NIH institutes, centers, and offices, so it is very much a collaborative group, and I see some of my colleagues from this group are on this call as well, as is Glen. Next slide, please. So this all came about back in about the April when the COVID started hitting. And one of the things I do is I listen in to the steering committee calls for each of the different consortia. And I was hearing the same thing over and over and over again. The first month I heard, how do our patients not get sick? What do we do? How can we take care of it? And then the next month's call, it was like, okay, this is not going away. How can I get my medication? How can I get my treatments? You know, should I go to clinic? And so the RDCRN, what we try to pride ourselves in is being shovel ready. for different types of studies, so they're able to act quickly. And within, I would say, 2 weeks, 3 weeks, they developed a survey collaboratively across the network looking at multiple rare diseases. And the targets of this group is, or what they're really trying to do, if you look on the left, is they're trying to understand what the impact to the families are. And anyone who's from 0 to 89 years old, I don't know why they picked that 89-year-old cutoff, and currently live in the US can participate. The survey is not limited to those with proven COVID-19 infections, but also collects information about how COVID-19 has affected the lives of individuals with rare diseases. So it's not just if you've gotten sick, it's what is it like not being able to leave your house, not being able to get your medications, a lot of other things like that. And this is not limited to the RDCRN. We've reached out to a lot of different communities you might have run across if you're getting any emails, or if you've ever been to NIH Rare Disease Day, you've probably gotten emails from us in reaching out. The project, the PI is Maurizio Macaluso, who's at Cincinnati Children's, along with Mark Rothenberg is one of the PIs in one of the consortia, and Thomas Ferkel in St. Louis is also one of the PIs of one of the consortia, and they all go across. Maurizio's at the DMCC. So they're all coming from different places and collaborating. And what we're really trying to do is look at not just the impact of an individual rare disease, because when this is over and people are making decisions about how are we going to deal with this if this ever happens again, we can't go 7,000 individual rare diseases at a time as to how we're going to deal with it. We need to look at rare diseases as a whole and compare amongst the rare diseases who might be more vulnerable for certain types of things. Next slide, please. So far we've collected, I say we, the PIs have collected 2,300 complete responses. Over 400 different disorders are represented. Right now, we have 17 different states that have over 50 participants from the state. So what we're looking at is we're really getting regional representation of COVID as it impacts rare disease. And as you can see, we're starting to hit a flat point on collecting data. We'd like to do something about that. Our target is 5,000 completed surveys. Next slide, please. So far, the demographics of the types of subjects we've been getting it's been primarily white. We're doing a lot of outreach and reaching out to different groups and organizations to try to help with that issue. We have primarily women responding or as respondents. Next slide, please. And so I asked them to look at what do we have for muscular dystrophy, because I thought you guys might be interested. And so far we have 6 people. And I said, could you break that down by the different types of muscular dystrophy? And so this is what we have so far. We welcome your groups to join us. Next slide, please. So the survey takes about 20 minutes. You can do it online. It's in a REDCap program. We have materials both in English and in Spanish that are available right now. The rare disease COVID, if you go to that site, you can complete the survey right now if you like, or you can contact Mauricio Macaluso or Jill Williams in Cincinnati. They can send you the materials, or you can reach out to me and I can send you the materials. And if you didn't write this email down quick enough, Glenn always knows where to find me. So we welcome participation from the muscular dystrophy community in this survey. And actually, if the groups want results, if you reach out to the researchers, you can probably pull out and do analysis on the muscular dystrophy information. Also, 90% of the participants have consented to be recontacted. So one of the things that they'd like to do is have another survey come out once this is done. And there is another group from Allergy and Infectious Disease that is interested in doing a serology study that's linked to this. So they'll be contacting the people who are participating in this study as well. So hopefully I caught up some time for you guys. And if you have any questions, just let me know.
Any questions for Tina? Thank you, Tina. We're actually doing extremely well with timing. So,
Tina,
yeah,
this is John
Professor,
Porter.
yes.
I'm sorry if I missed it and you said it, but what's the cutoff for the survey, the cutoff date?
The cutoff, they were originally saying July 31st. So
Okay.
there's a fair bit of time. So it could be potentially extended depending on, what's happening in the
Great,
COVID.
thanks.
Yeah. Thanks, John.
And Glenn has put the actual link to the survey on the chat box. So if you click on the little cartoon bubble, you can, at your convenience, write down the link to the survey. So hopefully you'll get some increased participation as a result of the meeting today.
And
okay.
Just
sorry, go ahead.
So if the family groups would like information, I'm going to put my email in the box. We can send the information in Spanish or in English. So if they want to put it on Facebook or tweet it out, that would be helpful. So sorry, and thank you so much.
No, thank you and gracias. So as you might recall, at our last session in November, we had a presentation about the FDA's patient-focused drug development program with a presentation from Meghana Chalasani. And we also had reports on Ph.D. meetings from Ryan Fisher, whom you heard from today, Paul Melmeyer, and John Porter. Today, we're going to continue this theme of patient-focused research with a series of presentations and discussion of patient-reported outcome measures. In today's session, we're going to learn more about this topic from the perspective of the FDA, from Michelle Campbell, who you'll hear from again, and Amanda Becker, who will provide the perspective of being a study participant. And Chad Heapwell is an academic researcher who develops disease-specific patient-reported outcomes, or PROs, and Lori Mignon, who will provide the perspective of the industry trial sponsor. So first, we're going to hear from Michelle again. So thank you, Michelle, for coming back.
Thank you. And so prior, as I said, I'm in the Office of Neuroscience, and prior to being in the Office of Neuroscience, I was on the clinical outcome assessment staff for 4 years, and also was the scientific coordinator for the COA qualification program. So outcomes is really where my background is in. And I think if we can go to the next slide. So the first couple of slides are actually going to be kind of a refresher to what my colleague Meghana Chalasani presented to you last fall. I actually spoke with her last week to see what she covered. And so I thought it might be good just a refresher to remind you of what she mentioned and how that work is going to really help impact what I'm going to be discussing when it comes to patient-reported outcome development and what we look for when we look at instruments to be used to support clinical trial endpoints from the agency perspective. So we have been doing patient-focused drug development meetings for well over 5 years now, and we have learned a lot. We know that patients are uniquely positioned to inform us on their disease burden and understand the impacts of available treatments. We know that our patients with chronic and serious diseases are their experts of what it's like to live every day with this condition. We've learned from these meetings that the chief complaints may not be factored explicitly into drug development plans. And these Ph.D. meetings that you spent your last meeting in the fall talking about, it really elicited patient input to better inform our clinical decision when we review things, when we get new drug applications come in, when we look at our benefit risk assessment. Because our patient stakeholders from the end of the first 5 years of this work was, okay, so you've learned all of this from us, but what is next? And so how do we really incorporate what we've learned from the patients into our next work? And that really is to how do we apply this into the benefit risk framework that we use when we're evaluating new drug applications and biological licensing applications. Next slide. So our next steps is that we have conducted a series of workshops and we are developing a series of guidance documents. Also, we affectionately call it the Ph.D. guidance series. And it is a unique guidance series, as not all of these guidances are written in what we would say traditional FDA guidance writing style. Particularly the first two guidances were really written towards our patient community audience and really in a way so they can be able to help contribute in the collection of patient experience data. So this guidance series is a four guidances in this series. The first two guidances, which are out in draft form, the first one is in collecting comprehensive patient community input on burden of disease and current therapy. This is out, and I believe we're working on the continuation of that to finalize. And this was really on more qualitative aspects of data collection. How do you talk to patients? Why is it important to talk to our patient communities? The second guidance that is out in draft form is development of a holistic set of impacts. So we're looking at burden of disease, burden of treatments, what is most important to patients. This guidance really is a continuation of the first guidance and really talking more of when you are systematically collecting qualitative data, how do we actually analyze it and put it together to help us really form and build that structure we need to get into how do we go from collecting this important patient information to getting into how do I develop or find the correct clinical outcome assessment I'm going to need to support my clinical trial endpoint for efficacy in a clinical trial. And so guidance 3 and 4 that will be coming out, they are not out yet, are really getting into that progression of now we're heading into that, let's get into that clinical trial. So guidance 3 is identifying and developing good measures for identified set of impacts going to be used in trials. We don't have the title, the official title yet. So this guidance in particular, if you are familiar with PRO development, patient-reported outcomes development, this will be the replacement guidance for our 2009 patient-reported outcomes guidance once it comes out. One of the limitations that we often hear about the 2009 patient-reported outcomes guidance is that it lacks all the other types of clinical outcome assessments, such as caregiver, performance measures, clinician-reported. Guidance 3 will incorporate all aspects of clinical outcome assessment development, and it will be used as that replacement. And then guidance 4 is going to be how do we go from this clinical outcome assessment to being able to support that clinical trial endpoint, and what is needed when we examine this. And we will also include the use of digital health technologies a little bit into this guidance. As I mentioned, and I can send this information out so you can disseminate it to the MDCC, is if you don't already have it from Meghana, is as I mentioned, all of these workshops were already conducted, but each of them have transcripts available, the slides, the agendas, and actually the video presentations, because it does give a good aspect of what some current thinking is and really some good methods as we are trying to put this all into writing. And if anyone has done a stint at FDA, you know how long it takes to write a guidance. And so this really is some good thoughts to think about while we're trying to get the rest of these guidances out. And I know Meghana mentioned this, but I just wanted to refresh your memories with this. Next step, please. Next slide, please. So I want to really get into patient-reported outcomes now. And so what do we really do? How do we go from that patient input that we get to we need a clinical trial endpoint? We need something to support that primary efficacy endpoint. Next slide. So as a refresher, we need to remind what do we define at FDA as clinical benefit? And we define it as a positive, clinically meaningful effect on how an intervention impacts. And so we say it's the improvement in how a patient feels, functions, or survives. And we know that survival is obviously very easy for us to be able to capture, correct? But the feeling and function is where we're going to be using our clinical outcome assessments most likely the most. And so we want to be able to describe this clinical benefit in labeling terms with that outcome of interest that was measured during that time. And so we always keep this in our mind when we are reviewing things is, will this outcome assessment that's being used in a trial, is this going to show an effect on how a patient feels, functions, or survives? And it's the constant thing that's always running in our minds as reviewers when we're reviewing our trial data. Next slide. So as a quick refresher, there are 4 types of clinical outcome assessments: patient-reported outcomes, clinician-reported outcomes, observer-reported or caregiver-reported outcomes, you may hear them as, and performance outcome measures. We, in collaboration with NIH, are part of the BEST glossary. So all of these terms are defined in the glossary, and I know this group is very familiar with that glossary. And we keep it up to date if we've made modifications, but I refer you to there. Next slide. For the purposes today was to really focus in on what a patient-reported outcome is. And so as a refresher, it's a measurement that can only come directly from the patient about the status of their health without amendment or interpretation of the patient's response by a clinician or anyone else. This includes pain intensity, seizure episodes, asthma symptoms, rescue medication, health-related quality of life as examples of concepts that the patient that we would consider can only really be assessed by that patient. And we want to make sure that patient-reported outcomes run the gamut. So it can be a single item such as pain intensity on a scale, or it can be complex measures like health-related quality of life. But again, it goes back, who is that reporter? And that is the patient. Next slide. So I wanted to spend a couple slides talking about a concept called fit for purpose. So I mentioned clinical benefit, right? So improvement of how a patient feels, functions, survives, we look at. The next thing we look at when we're looking at a clinical outcome assessment, including our patient-reported outcomes, is really, is this instrument what we call fit for purpose? And really is, and this is the definition that we use, is that we can conclude that an instrument is fit for purpose is a conclusion based on the level of work that's associated with the tool is sufficient to support its context of use. It's sufficient to say, in this patient population, measured this way, I can conclude that this patient-reported outcome is assessing, for example, let's say pain in disease X in this setting. And so we really will use the term fit for purpose. Is it fit for the scenario that we are studying? What is our trial design? Does it make sense with what the drug's mechanism of action is? All those things pulled together, is this the correct clinical assessment to be used? So next slide. So can you go back one, please? So when we are reviewing and having submissions come in for review in the IND stage, when we are looking at what is being used, and this is the same, it could be a patient-reported outcome, it could be a clinician-reported outcome. We think about this in our heads while we're reviewing things. So the first thing is this context of use concept. Is it being used in the appropriate population? Is it the actual population that the study is meant to be? What is the study design? Does it make sense? Is it appropriate? Are the measures, have they been validated and are they reliable? So are they important to patients? And so so are we measuring what's important to a patient? At the same time, does it make sense that what's important to patients we expect to see a change in based on what the treatment is? And I know that's a fine balance that sometimes people struggle with, because what patients may be important to patients, we know the drug mechanism may not target. And so we have to find that balance of making sure that the drug development program and what we expect the drug will do on the body also correlates to still what's important to the patient. And so we have to really find that balance to make sure that we are measuring the appropriate thing, so we will see if there is an actual clinical benefit. Are the concepts clinically relevant? Again, it goes to that balance I'm describing. Does it make sense? Can we see it? How long do we expect to see? How long will it take for change to see? So are we measuring the concept appropriately and often enough to make sure we will see change? And we also need to make sure that we can communicate in labeling in a way that it's accurate, interpretable, and not misleading. And this is where you'll hear the term well-defined, which is what is defined in our Code of Federal Regulations for us. Next slide. So why is it important to really take the time to make sure we have fit-for-purpose clinical outcome assessments? Because we know that if we use inadequate instruments that are not fit for purpose or not fitting that appropriate population, and this could be for what we call off-the-shelf or existing instruments. This could be instruments that were modified. This could be instruments that people have developed. If we're not using fit-for-purpose instruments, it can impact our clinical trials. So, for example, we could have a lack of patient-centered instruments, so the tool does not assess what's important to patients. And so we may be missing something from the trial, from the patient perspective of capturing what's important to them. We could have what we call content validity problems, where the instrument's content does not accurately assess the target concept of the population. Again, that's going back to understanding what are you trying to show improvement in or slow progression of in your disease, and so what aspects of that. So understanding that disease is important. Do you have the right severity of population to make sure you can actually see that change? That could impact if we don't have that. And so a trial could fail, but it could be because of the instruments. It was not sensitive enough, which comes to the third point, which is the ability to check change. And that you may not be able to detect a treatment effect because maybe we have the wrong population there. The instrument was developed for a more severe population and we have a milder population. Or it's just the wrong instrument altogether. And so I think we would rather have a trial not be successful because of something in the trial, maybe the molecule or such, instead of the instrument. And so that's why we really make sure we're wanting to select as closely as possible what we call these fit-for-purpose instruments. And that's why we encourage early communication with us to have those conversations if we're working in disease areas where there may not be prior information available. And so we have to think about what may work and have those discussions. Next slide. So I'm throwing up here as a reminder, this is our 2009 guidance. So while I mentioned that we are going to be replacing it soon, this is still, though, our current guidance and our current thinking. And so it does describe how we define and interpret well-defined and reliable, which is per our regulations. And that COAs, this guidance really describes good measurement principles. But we also want to acknowledge that we have to provide flexibility in judgment when practical demands are needed. So particularly in our rare disease space, we have to be flexible because creating a de novo instrument probably is not going to be the best thing to do with the timing of drug development and the numbers of patients you need. So we are open to flexibility, and so that's why this early engagement, we encourage this because we can have those conversations and determine where flexibility may be needed, what needs to be done early in trial design to benefit the trial itself and the ability to recruit patients, and also the data that's coming in so we can interpret the data will be a lot cleaner. Next slide. So when we are looking at an instrument, it comes in, and we're trying to say, you know, what does this instrument tell us? We want to look at concepts of good measurement principles. And so what you see here are, these are the questions that we will ask ourselves. Is this an appropriate instrument for the population? You've heard me talk about that a lot, the context of use. Does it measure what's important to patients? This is the content validity aspect of an instrument. Is there multiple domains or concepts being measured? Are they overlapping? I think the big things that sometimes we have some of the most discussions on is the instrument sensitive to change? Is the change clinically meaningful to patients? And so what does an improvement of a score mean? And that gets into that clinically meaningfulness. And so these are things that we think about as reviewers when we're getting our packages and are coming in and thinking about the appropriate clinical outcome assessment, PRO, to support that trial endpoint. Next slide. And yeah, thank you, Emily. So this is a little bit more in detail about when we're evaluating and when our industry colleagues may be putting together their trial designs and they're thinking about their measurement strategy, which should go hand in hand with their trial design. These are the concepts and things that they may be thinking about. And so I've talked about the first diamond there on target population and measurement concepts. So a little bit, but then we want to make sure that we have where is the COA that's being used and the endpoint I want to support, where is it in the endpoint hierarchy? How do we define it? Is this the appropriate, is it in the appropriate, if it is a hierarchical situation, is it in the appropriate location? Should it be elevated? Should we move down? Are you allocating alpha when the study's happening? So we look onto this. Typically for our primary endpoints, as the name implies, these are endpoints that are going to be used really to establish effectiveness for approval. Our secondaries are extremely important. They are typically supportive of those primary endpoints in the trials and help, enhance and support that trial data and information when we're making decisions. And so we will look at that, and we may make recommendations to move endpoints based on what the entire development program looks like. And then the big thing is also that scoring and interpretation. So are you weighting a scale, for example, which means I can't interpret it, and how do I describe this? I like to think of this as not only do we have to describe this into labeling, but how would a clinician or how would we explain this to someone else, what that score change would be. And so we will look at how scores are being developed to make sure that they are interpretable in ways we can describe. Next slide, please. So characteristics we look at when things are evaluated, we look at content validity. That's the first piece. And so typically that is where all your qualitative work is done, your literature review, things of that nature. And so we will look to see if content validity has been established. And it's important to note that if content validity hasn't been established, no perfect way of the quantitative side to the psychometric work properties can overcome a lack of content validity. So if you don't have content validity, you can't try to correct for it in another way quantitatively. It just will not be overcome. And then when we look at our psychometric properties next, we look at reliability. We look to see how it performs over time. We want to know, does the instrument have an ability to detect change, so longitudinal. So if we're lucky and we have a phase 2 trial, this is really that opportune time to test out instruments that may not have been used in this patient population before to see how they perform. So there's no really surprises come phase 3. That opportunity does not always happen in our rare diseases, and we recognize that, which is why that early engagement is important to have those conversations. And then I would say the most important thing after that is interpretation of clinically meaningful change. And specifically, we're talking about clinically meaningful with inpatient change. So not between groups, which is what we use when we look at efficacy, but what is going on within a patient is really what's important to us. And so if you go to the next slide, please. And so I think we're hearing more about clinically meaningful change a lot more. I think I'm talking to more groups these days about how do we establish clinically meaningful change from instruments, and the peer guidance describes quantitative methods of how to establish meaningful change. But I think it's important to think about it is that we know that statistically significance alone is not sufficient. We need to make sure that changes have to reflect the positive clinically meaningful effect of intervention. So that clinical benefit, which is why I started with it. So are we improving how a patient feels, function, or survive? So an example could be, well, I have a positive study, right? It's statistically significant, but is that 3-point change really clinically meaningful? And so that's why we really need to assess all of the aspects of the instrument and what was done and how it was assessed and its ability to check change. And this is, you hear more discussion about this is when we have small amounts of change, right? I'm using 3 points as an example. Let's say it was 3 points on a 100-point scale. Well, that doesn't seem maybe meaningful. Maybe it's meaningful if it was a 10-point scale. And so this is where these discussions happen when we may be having smaller benefits. Obviously, if we have something that's really big differences between placebo and treatment, I think that's obvious to us. And so it's important to remember that our primary endpoints in our pivotal study should be designed to reliably identify meaningful changes in how a patient feels, functions, or survives. And so, as I mentioned, the PRO guidance talks about ways that we can do this quantitatively, statistically, doing some basis. But we should always think about in our heads is, does the assessment measure or reflect something of a significance to the patient? And does the scoring of the assessment also reflect clinically meaningful changes? So we need to think about this throughout the entire design of it. And it really, again, is important, and this is why I cannot encourage enough, next slide, this early engagement. I know some people think, I don't want to come to the FDA until we have everything ready, to have that conversation. But we really do mean it when we say, please come talk to us early. We know this is a critical, point can really help this early engagement with us. And so we encourage the development, implication of patient-focused clinical outcome assessments in our trials. Again, this early, come talk to us early. And that clinical outcomes used to support clinical trial endpoints should be clinically meaningful and translate to an improvement on how a patient feels or functions. And so that is my presentation. I thank you. I will take any questions. I know we're going to have, I think, a Q&A after all of the presentations. But if there's anything specific, if there's time, I can take a question now.
Thank you, Michelle. I just had one quick question for you. You were very emphatic saying that patient-reported outcomes are reported by the patient only. But what happens if the patient is a child? Because actually our next presenter is the parent of a patient. So does that extend to a parent?
So it's a great question. And obviously, this happens not only in our pediatric populations, but maybe our older adults where there's cognitive impairments. Or cognitive decline, we run into this as well. And that's why we have that observer or caregiver reported option. It's slightly different in the PRO. So we want to make sure that in the instance when we use the caregiver or observer reported outcome, it is things that another person can observe and can see. So the classic example we would say is we would not want the parent to report on their child's pain, because while they may physically be crying and we think that they are probably in pain, they can't actually, if you had to give a value, say, this is how much pain my child is in, and the child can't self-report. And so I think that's why it's really important to come have those conversations with us when we are getting to caregiver reported outcomes, because they are very important, and we use a lot of them in our rare disease, in pediatrics, in adults with cognitive impairment. And so I would say there in particular, come talk to us early so we can make sure that the question is going to capture correctly what we need. And it can be done successfully. It can be done, but there's some nuances that we just have to make sure are being taken care of with that.
Okay, great. Thank you. So I think we will move on to our next presenter, who is going to tell us about this issue, because it's Amanda Becker, who is mom to Jack, who has Duchenne muscular dystrophy. And she's going to tell us about their experiences participating in a clinical study collecting patient-reported data. So, Amanda?
Yeah, can you hear me okay? I had a question for Mr. Campbell on the previous discussion that I sent to Chad. Maybe we can address that after this presentation. Thank you.
Okay, can you hear me okay? Oh, hello?
Yep, we can hear you, Amanda.
Yes, we can hear you. We can hear you, and we'll address Daniel's question later.
Okay, thank you so much for having me. My name is Amanda Becker. I'm a mother of a young man with Duchenne muscular dystrophy, and I wanted to thank you for having me today and forgive my technical skills because I'm not in the world of webinars, and you'd think I'd have learned more by now in being isolation here. But I have been in the Duchenne space for about 14 years, as my son was diagnosed when he was about 1. I'll move on to the next slide and show you a picture of our family here. That's my daughter, who is a high school senior, so she has graduated. We've done the whole virtual high school graduation, and looking forward to trying to get off to college in the middle of a pandemic, which has been a challenge. I live here in New England, and I've supported a number of Duchenne foundations over the years. I've tried to be an active community member, realizing that when Jack was diagnosed, there was really not a lot going on in Duchenne. It felt like a very bleak landscape. There were no human clinical trials. Quick Google searches and came up blank with absolutely nothing. So it's inspiring for me to participate today and see all of the progress that has been made, and especially thanks to the Muscular Dystrophy Coordinating Committee and the MD Cares Act and all the investment that's been made in infrastructure to try to help this disease, as well as all the muscular dystrophies. So I am just a volunteer, and I volunteer for a couple of different organizations. I have volunteered frequently for PPMD, Parent Project Muscular Dystrophy, and I have tried to attend a number of the advocacy conferences in Washington, which I'm grateful to all the members who have attended those meetings over the years and try to help educate us as parents and help us advocate for what we need in order to move this disease forward. So I am grateful to be invited today. I've tried to follow the research. As I said, when Jack was first diagnosed, it was just shocking with the severity of Duchenne and the fact that there were no treatments. There was real lack of trials. It was just bleak. So that puts a parent right on the bandwagon of following the research and seeing what needs to happen to, try to find a treatment or their cure for Duchenne. So that's just a little bit of background about me. And next slide. And I have been an observer of the many things that the presenters have talked about. A lot of it's over my head, to be honest with you, but I have been aware of the patient-focused drug development aspects and the importance of incorporating the patient voice. And I guess the story that I have to share today is just my experience of having gone through early clinical trials in Duchenne and watching the field develop and evolve. So one of the things that is common in Duchenne is the PODC, the Pediatric Outcomes Data Collection Instrument. And this is we've been asked to fill this out several times. Just to take a step back, Jack was in some earlier clinical trials. He's now 15, and I would say this is going back 8 or 9 years that we were able to participate in a clinical trial. And the PODCI was one of the very first tools that was used in the trial that we were in, and it is also currently used in standard of care visits. So we've filled this questionnaire out several times. And the first comment I'll make about this tool is that it is the most common, one of the most common tools used in ambulatory trials in Duchenne. One of the concerns that, and I guess my presentation today is really just to give you the patient perspective and just share personal stories about what we go through as patients and families filling out these tools, and what the, you know, just some of the thoughts that go through your mind as you have this experience. And I think one of my major points is that I value everybody on the call today who's trying to make these tools as valuable as possible, as accurate as possible, and they value the quality of the information that you're able to gather for this whole process. So my first comment is about the, you know, the ambulatory nature of the PODCI, and how most of it has really focuses on ambulation. As we know with Duchenne muscular dystrophy, that ambulation is only part of the story. So we are challenged in the community, and the community is very divided sometimes between ambulatory and non-ambulatory populations, when actually we want to include all patients, young and old, and anybody who's fighting Duchenne, no matter their severity or their age or their stage. We need tools that would help us, you know, defeat this disease. So the first slide, next slide, please. So I think just a few comments. We've taken a few samples of the PODCI tool, and this is just a sample of some of the questions that families are asked to fill out when you go to a visit. And again, it could be a trial visit or it could be a clinical visit. And just to put it in context, you're often arriving at a busy children's hospital. You've had to park your car, locate your parking ticket, or save it for later. It's often kind of a confusing and overwhelming time, especially as you are arriving with a child and trying to keep that child busy or occupied while you go through these tools. So again, just, you know, the accuracy and the value is important. So for the PODCI, for us, it was some of these questions are really hard in the context of Duchenne. A lot of these questions seem very basic and seem very obvious and seem very simple to someone just reading them, but as a Duchenne parent, all of these really sort of pierce your heart, because none of these tasks are easy for a child with Duchenne if they are able to do them at all. For example, running short distances, a Duchenne parent just cringes at the thought of watching their child even try to run at a young age. It's usually very awkward. It's usually very disparate from what a typical child can do. The same with riding a bicycle or tricycle. You'd think that's just a given for any child, but with Duchenne, it's a heartbreaker to think that your kid may never ride a bike or ride a tricycle. And in our family, we bought several ride-on toys and could never actually find one that would work very well. Climbing 3 flights of stairs, that jumped out at me as taking these surveys, because I would, knowing and understanding Duchenne, you're afraid of stairs. You're afraid of falling down the stairs. You're afraid. I would never ask my kid to try to climb 3 flights of stairs. So just my point is, as a parent, when you're filling this out, you have these fears of my kid falling down a stair, or that time he did fall down the stair. Those things pop into your mind. Walking 3 blocks, I recall taking this and thinking, did they mean a city block? Did they mean just a little block? I live in a very small town, so our blocks are very short. So I didn't really know, is that like a Washington, D.C. block or New York City? These questions were hard for me to put into context. Again, getting on and off a bus, it seems like a very simple question, but as a Duchenne parent, it's a heartbreaker to think that your kid can't get on the school bus. And the very first day of school and all those pictures that parents take, just having their kid go up those bus stairs is just I can't tell you how heartbreaking it is as a Duchenne parent, you know, to try to get your kid on the school bus, and it's a big challenge. The next slide, please. This next portion of the PODCI was challenging for me. because as a parent, I was always trying to put up a facade, if you will, to my child. I would say, oh, of course you look great. Of course you're happy with, you know, what your body can do today. So some of these questions I found hard to answer because the other thing is I felt like these are a little bit more appropriate for a teenager. When I was taking this when my son was 6 or 7 years old, these weren't his top concerns about how he looked or happiness with his body or what shoes or clothes he could wear. I mean, at that age, you know, you just want to get your kid out the door with clean clothes, and are they in fact wearing shoes would be a good thing. The ability of the child to do the same things as his or her friends, that is a heartbreaker for a parent with Duchenne, because many things are so far off from what might be typical. So a lot of these questions you can understand as a parent, where if you have a typical child, these questions might make sense. But again, within the context of Duchenne, which is progressive, they felt off the mark to me. I felt like this tool didn't capture muscle weakness, which is the primary symptom of Duchenne and in many muscular dystrophies. So there was really no measures of how weak is your child. As a parent, then back to the other questions, can your kid run? Can your kid walk? Well, my kid, he might be able to walk, but he's too weak to walk on a beach. He's too weak to lift up a baseball bat. He's too weak to pick up his toys. He's too weak to get on and off a swing. I mentioned the swing because some of these surveys say, can your child participate in recess or can they participate in gym? And these, you know, my kid is able to show up and sit on the sidelines. It's very, very difficult for him to participate in anything that's not adaptive. So turning back to the slide where it says, can your child participate in pickup games or sports with other children the same age? Really in Duchenne, most children have to use adaptive activities, adaptive sports. When the children were young, they needed an adult support or a sibling or a friend to help them play soccer, or I'm laughing now as I see jump rope or hopscotch. You know, some of these things that are just so very typical for most children really don't even tap into what the child with Duchenne is facing. Next slide, please. And this slide for us as a Duchenne family, a lot of these, again, didn't match the expectations for what we would hope to see with treatments. And when you're in a disease that's progressive, it's severe, and there is no treatment, just the question of what expectations do you have for your child's treatment, I would sit there and just kind of hold my head and say, you know, these are really tough questions, because I don't feel like they're even assessing my kid's disease. And that can be hard on a parent as you take these tools during a visit. You really want to feel like your time is valued, your input is valued, and that you're able to give good information to the clinicians or to the researchers. The families are investing a lot of time in attending the clinical trial visits. I'm sure this group has a great appreciation for all that goes into planning the trials and the commitments that the families make to try to go. And when you feel like the tools aren't necessarily capturing what's really important, it can be frustrating. And again, as a parent, there's an extra stumbling block because, you know, there's the emotional piece of wishing that there was a treatment for your child. So having and I felt like this tool didn't really reflect the progression of Duchenne or the urgency of Duchenne. And then the final question, I'm just looking back to some of these other questions, they really just didn't apply to my son at a younger age. To be able to do more sports, well, he really couldn't do much. To be free from pain or disability as an adult, that's a loaded question for a parent of a child with Duchenne, as you're just hoping that your child survives to adulthood. Then the final question, if your child had to spend the rest of his or her life with his condition as it is right now, it's again a very emotionally taxing question for Duchenne muscular dystrophy, where it's so severe. Next slide, please. So I guess just in summary is just my reflection on this tool, that it didn't really reflect the severity and the progression of Duchenne. In terms of quality of life, there were questions lacking reflecting the quality of movement and the quality of his life. Some of the physical activities that Jack was able to do, say he could go up and down a step or he could walk those 3 blocks, it didn't really reflect the quality. So we would take these surveys year over year, and I would know that yes, he can still walk 3 blocks, or yes, he can still walk around the perimeter of the grocery store, but the quality of that walking, the time it would take for him to walk, the risk of the falls, and when you're talking about things that are meaningful to a child with Duchenne, or anybody really with muscular dystrophy, you know, the number of falls. How many times is that kid falling when he tries to walk the 3 blocks or tries to walk around the grocery store? They might be able to do it 1 year without falling. Another year, they would fall repeatedly. So number of falls. And the point that I spent time tracking things on my own, being an early observer of some of the trial development in muscular dystrophy, I knew that we didn't have very many tools to measure progression and measure potential therapies. Notably, the 6-minute walk test, we all know that that is only applicable to a certain subpopulation, and the majority of muscular dystrophy patients can be left out of that. So I added a few things that I would talk to my clinicians or the trial folks about. One of them was the falls. I said, why isn't anybody counting the number of times my kid falls? That really is important when it comes to losing a tooth or breaking a bone or fracturing your femur. I also mentioned getting in and out of the car. That was a big thing and still is for many families with Duchenne and anybody with a physical disability. It's a real obstacle for being able to just get where you want to go, and the level of assistance that you need to go somewhere, or the level of equipment and adaptations that you might need. So that's was a big thing for me, and I actually noted on my trial paperwork, and the trial folks actually noted it as well, and we would talk about how is it easier or harder now to get into the vehicle. So it was a stressful experience. I think my advice to other parents when I'm talking to newly diagnosed parents, or I'm talking to parents about participating in clinical trials, I say that you know, managing your stress and managing your expectations and planning the time. I always appreciate going to a visit or a trial visit or even a clinical visit when people say, we have some things we want you to fill out. It's going to take about 10 minutes, or it'll take 15 minutes, or it'll take 2 minutes. So just helping families set those expectations, and also being able to do it. If you arrive at an appointment and there's a lot going on and you don't have time to fill out all these things, that you feel badly because you want to be giving the best possible data and information that you can, but it could be a busy clinic day, it could be a busy hospital day, and things can get lost. The fact that it's an emotional minefield, it really can be, and I think over time you can expect when you go to a visit that some of these questions will throw you off, and personally I found myself sometimes getting really stuck in feeling like I was answering questions that didn't necessarily relate specifically right to my child. So I was hoping for the best and hoping that it would be helpful. And I just want to end on saying thank you for all of these efforts. And the speaker prior to me I just couldn't be more grateful to hear people talking about finding endpoints and measurables that are clinically meaningful and meaningful to the patients. And I think that caregivers and patients they're highly accurate. I could tell you the day Jack couldn't access the playground, the day he could no longer get off the floor, the day we had to buy a new car because nobody could lift him in any longer. So we can have often usually pretty accurate information to share, and we hope that our information is gathered and valued and is able to be used by the researchers as best possible. So that's the end of my comments. I'm happy to take any questions. And again, I appreciate all the time and effort of this committee and all of the efforts going on today.
Thank you, Amanda. Are there questions for her? I think you very eloquently presented the case for why no research should be done without input of the people or their immediate family who are affected by the condition being studied. I mean, just the concept of getting in and out of the car, I mean, to think of how important that is, and that's not captured in the questionnaire. So you just, went point after point and explained to everyone why the direct insights from people such as yourself or Jack, now that he's a teenager, are critical to getting the right questions to pinpoint the functional aspects of the condition being studied. So thank you very much. I'm looking at the chat box specifically for questions for Amanda, and I don't see them. And now we're running a little bit behind. So I'd like to thank you again, Amanda. And I'd like to go to our next presenter, who is Chad Heatwall from the University of Rochester. And Chad has developed several patient-reported outcome measures for neuromuscular diseases. So hopefully he is going to be able to answer some of the issues that Amanda raised with his presentation. So Chad, the floor is yours.
Thank you so much. It really is a pleasure to be here with you, and I really appreciate Amanda's comments as well. So I'm a professor of neurology at the University of Rochester, but also a graduate of the NIH Experimental Therapeutics Program in neuromuscular diseases. And today I'm going to chat a little bit about disease-specific patient-reported outcome measures and how these measures can be used to really optimize the measurement and quantify the measurement of how a patient feels and functions. Next slide. Those are my disclosures. Next slide. So why do we need disease-specific patient-reported outcome measures? As Amanda mentioned, and Michelle as well, there are certain things that only a patient can tell us. And a patient has the opportunity to provide a unique window into the effects of disease and treatment effectiveness that we can't otherwise gather through other mechanisms. Disease-specific patient-reported outcome measures are also thought to be more responsive to small but clinically meaningful and relevant therapeutic changes. And therapies should be shown and proven to have relevance to a patient community. And a PRO is really a way to do this. Next slide. When developing disease-specific PROs, there are 9 characteristics that we strive to incorporate into all of our instruments. And these are listed here before you. So first and foremost, the instrument has to be meaningful to a patient population. It has to be reliable, has to be able to detect change or be responsive, have a good response range. It has to have good statistical inter-item correlation, recall period. It has to have limited patient burden. You can't ask abrasive questions to a patient or a patient community. There has to be no redundancy in the questions. And most importantly, it has to be valid. And there's about 7 different ways that an instrument has to be valid. I won't go into them today. Next slide. We started developing disease-specific PROs 15 years ago. And one of the things I did was working full-time. I went back and got a master's degree in psychometric methodology and clinical investigation, working with members of PROMIS and Neurological to really identify this need and to develop instruments for specific populations. And since then, my lab, we have many individuals that are participating in this and part of our group, including epidemiologists, biostatisticians, patient advocacy groups, psychometrician, linguists, neurology patients, and then also research partners throughout the United States and throughout the globe have helped us with the validation process. Next slide. To date, we have 62 completed instruments. These are being used throughout the world, and we have 40 additional ones in progress. These instruments have been translated and validated into 10 separate languages. They are patient-reported. We also have clinician-reported, observer-reported, age-appropriate forms, including pictorial-based forms for pediatric populations. We have short forms and also clinical assessments. This is a partial list of the diseases. We really started out with the muscular dystrophies, and we've since broadened our efforts to include other neurologic conditions and even diseases outside of neurology. But the instruments we have the most experience with are myotonic dystrophy and FSHD, and we're doing limb-girdle, we're doing IBM, and we're also Duchenne's muscular dystrophy. And I can guarantee you that we'll have a question about getting in and out of a car. Next question. Or next slide, please. So these instruments are being used by a variety of organizations, mostly academic researchers who are running therapeutic trials, but also pharmaceutical companies. The FDA has sponsored the use of this in one of their therapeutics trials. Foundations and registries are also utilizing these instruments. Next slide. Next slide, please. So why disease-specific instruments? There are other instruments that are available generic, and there's also the possibility of doing repurposed instruments. Well, disease-specific instruments have a higher relevance and content validity for target populations. They have an increased sensitivity to detect significant therapeutic change. They have a higher precision, simpler application, lower burden to the patient, and they can emphasize the relevant symptomatic themes of a specific population while excluding non-relevant issues or questions. Nothing is more abrasive than asking a patient who's in a wheelchair how their marathon time is. They can be scored to prioritize high-impact issues for a specific population, and overall, they are best suited to measure disease progression or therapeutic gain over time. There's significant literature that shows this. Next slide, please. I'll talk briefly about one of our first instruments. It was the Myotonic Dystrophy Health Index. There's been 7 validation papers for this particular instrument. This is for DM1, and we developed this to extensively satisfy the criteria that was published by the FDA in 2009. Every time a patient completes this instrument, it's made up of subscales. So they get a total score and also 17 subscale scores that represents the areas of greatest importance to this population. There's over 100 questions, but really it's only one question. How much does the following impact your life now? And a list of items. And it takes on average 12 minutes to complete. We base this based on a cross-sectional study of 278 patients. We've shown it to be reliable. The instrument can differentiate between known groups of patients with more disease severity. It correlates to functional measures. It has extremely low ceiling and floor effects. We've extensively beta tested this to show that it's approvable and acceptable to patients. It's been translated and validated and used in English, Japanese, Italian, French, French-Canadian, German, Dutch, and Swedish, and is being used in therapeutics trials throughout the world. Next slide. So based on the data, the NINDS common data elements identified it as the only PRO that is highly recommended to measure multifactorial disease burden, and one of only 4 instruments that's highly recommended in general. Next slide. Every time a patient measures this, it's composed of subscales, like I mentioned, and the subscales have variable amount of questions, and the questions in the subscales are weighted based on what patients feel is most important. And so while fatigue or pain or mobility would often be relevant to many muscular dystrophy populations, it should be said that the relative importance of these questions and these concepts is different between these populations. So every time someone completes this, they get a score at each one of these areas, and you can read this here. Next slide. So a lot goes into this. So this is a general overview of how these instruments are built. They're based on qualitative interviews, large-scale survey initiatives, expert opinion, literature review, advanced statistical methods. We utilize input from lieutenants who participated in the PROMIS and Neurological. And these instruments are used not only for clinical trials, but also for patient monitoring in the clinical setting, as a guide for development of other instruments. And really the ultimate goal is by having these instruments available to the research community to improve overall patient care in drug development. Next slide. So how do we know these patients represent how a patient feels? Well, we ask them. So we start out with patient interviews. We collected over 1,000 quotes. We identified potential areas of importance to this population, so 221 symptoms. And then we validate this using a large cross-sectional study, in this case, 278 patients. We've conducted other studies that have approached 1,000. So we get a relative importance and also relative prevalence of each of these issues. Next slide. Then we go back, and after we develop the instrument, we ask patients about it. And I think after doing this with many subsets of patients, including those in myotonic dystrophy, there's 2 points that come out. Is per patient, the most important aspect of a symptom is how it interferes with the patient's life. So this is a critical importance, and our instruments do our best to represent this. And I should also say that the frequency and or severity of an individual symptom is irrelevant to patients unless it has a clear impact on a patient's life. And so these 2 points are really what patients have told us over the years. Next slide. So how do we know this represents how a patient functions? So this is a known groups validity graph. And what we see is MD high scores, and that goes from 0 to 100, 100 being the greatest disease severity, by employment status, genetic repeat length, education status, and duration of disease. And what we see is the instrument is able to differentiate between groups of patients with more or less disease. Next slide. And when we do validation steps, we do it for each of the subscales as well. And so these are the subscales of the Myotonic Dystrophy Health Index. And what you can see is that based on those that are employed versus those that are not, or those that are disabled versus those that are not, the subscales are also able to perform well and differentiate between those with more or less severe disease. Next slide. Also, we've correlated MD high subscale scores and total scores to standard functional measures. It has to represent how a patient functions. We've done this 3 or 4 different times in different studies. This is the latest data through the Wellstone supported MD-CRN study, 113 patients at 6 research centers. And what we've seen is that there's very high correlation between MD high scores and strength, ambulation markers, breathing markers, and disability markers. Next slide. We also wanted to compare the usability and use and performance of our instrument versus generic and semi-generic instruments, including the SF36 and the INQUAL, which is a semi-generic instrument. And here we asked patients, in this case 50 and 52 patients, which instrument was most relevant to you. And you can see that they preferred the MD high over both the INQUAL and the SF36. Next slide. We've also asked them questions about meaningfulness of the instruments, perceived responsiveness, perceived usability and patient burden, and overall performance. And across the board, the MDI, despite being slightly longer than these instruments, is widely preferred over the generic instrument and the semi-generic instrument. And this really makes sense because the questions and items were specifically selected for this population. Next slide. But how does it perform? So again, we looked and compared the comparison of these 3 instruments and being able to differentiate disability status or employment status. And the instrument that was able to do that most effectively was the disease-specific MD high compared to the INQUAL and the SF36 represented by the SF60. Next slide. We also compared it to functional measure. Can it represent how a patient functioned? We picked 3 standard functional studies, the MMT or manual muscle testing, 6-minute walk distance, and muscle impairment rating score. And in all cases, the most solid correlation between those functional measures was the disease-specific instrument. Next slide. And of course, we have the standard metrics for all of our instruments so that they can be used to design future clinical trials. We've longitudinally tested our instrument to develop and determine the minimal clinical important difference, or MCID, using anchor-based methods. It's about 8 points. We also have test, retest reliability. We have standard deviation of the instrument going forward. And we have specific metrics based on demographic information of the patient population. And all this data is really essential for planning, empowering, and determining sample size based potentially on these instruments. And there is a sample size saving. As with any study, it depends on what type of effect you're looking. If you use the MCID for the MD high, you at the very most need 36 patients to determine a clinically important difference. If you want to use a higher treatment effect, but even less than the difference between those who are disabled versus not, you can go down to as few as 10 patients. And so this is a really responsive, powerful way that is meaningful to patients and can really observe meaningful changes in disease burden. Next slide, please. So in conclusion, disease-specific health indexes, when developed correctly and properly, provide a valid and powerful mechanism for serially monitoring the multifactorial disease burden of patients with muscular dystrophies and other diseases, and represent both how a patient feels and functions. Next slide. And that's all I have. Thank you very much for listening.
Great. Thank you very much, Chad. Walter, you had a comment. Would you like to make the comment and see if people want to react to that?
Sure, and maybe Ryan could help. So in terms of the use of the items and the scales, I would say that you don't want to be wedded to a scale. You want to be wedded to the question and use the scale that helps you answer the question. So Ryan mentioned disease progression as being the thing that they were after, and I always thought that if you really know the disease, you've seen people go through it, whether it was a parent or a doc, that there are certain stages of the disease that just keep coming over and over again. And those are the kind of questions you want to put into your PRO to get a sense of where, what stage of the illness has the patient progressed. And then the benefit is if you have something that, really precise, you can measure differences for a certain period of time that is conducive to doing a clinical trial, and then you have a real instrument. So with Ryan, I guess the question is, do you on how your scale changes over time, and what's in terms of doing these clinical trials, what time frames you have to follow people to see a detectable change?
Yeah, we do. I think that's a very excellent point. And for many of our instruments, we have longitudinal data that we've either conducted ourselves or our academic partners at other institutions have, or even pharmaceutical companies who've implemented these into therapeutic trials have provided us. And we select the questions for them to be as most responsive, but also most relevant to the patient population. And I'll give you an example that I think hits on what you were saying. So we do have an instrument for patients that have spinal muscular atrophy, for instance. And this instrument is designed for patients age 12 and up. And so we have evidence and data based on SMA type, and that the instrument can actually differentiate between SMA type in that particular population. And as far as disease progression, it depends really on the disease you're talking about. A lot of these muscular dystrophies are slowly progressive, especially in the adult population. And so there may be some subscales that progress on a faster, at a faster rate than other subscales. And so we do have data on that as well. And, you know, the thing that can be noted is that, you know, not all therapies will affect every symptomatic domain. And so a therapeutic agent or a pharmaceutical company might come in and say, hey, I have a drug that we think is going to help mobility. And so they can focus on the subscale for mobility. Or they might come in and say, I have a drug that's going to help pain, or a drug that is going to help any number of the diseases that our multifactorial instruments can measure. And so in those cases, you know, we can look at those individually and we can see, you know, which ones is progressing faster. And that can help really help select therapeutic clinical trial design and have a better understanding of how the outcome measure should perform and would perform in the case of a therapy that works.
Great. Thank you very much. I think we're going to move to our final speaker, who is Laurie Mignon from Ionis Pharmaceuticals. She's going to tell us about their experience using PROs in clinical trials for myotonic dystrophy and other conditions. So Laurie is Laurie Laurence or Laurie? So I guess we'll hear the minute Laurie starts speaking.
Thank you, Dr. Bianca. It is Laurence Mignon, and I'm a director at Translational Medicine at Ionis Pharmaceuticals. I was in the clinical development department at Ionis for the last 5 years and have been involved with our spinal muscular atrophy and myotonic dystrophy. Next slide, please. So just my disclosure, obviously an employee and stockholder of Ionis Pharmaceuticals. Next. I just wanted to give you an overview of how I have organized my talk, and I think you'll see that a lot of the items I'll be talking about is really things that both Michelle and Chad have touched upon, even Amanda, but really kind of just from a different perspective. So I'll just go over patient-reported outcome selection for clinical development, and then if you can go back, please. And then we'll talk about myotonic dystrophy. Thank you. And then the role as PRO, or the potential role as PRO in a post-approval setting, which I think is also quite important to consider. Next slide. So I wanted to start out by giving a perspective of the number of drug approvals that include a patient-reported outcome in the label. And so there were really 2 recent papers that were published in Value Health that have focused on that topic. One focused more on orphan drugs and the other focused on drugs in general. And so if you focus your attention on the graph first, this is from the 2017 paper by Hong that specifically focused on orphan drugs. So the blue bars represent the number of orphan drugs that have been approved. The hashed bars represent the number of orphan drugs that also include the PRO label claim. And then the line just shows you the proportion of this. And so I think it is obvious to see that starting with the draft FDA guidance in 2006 and then 2009, that there was an increase in the number of PRO label claims in approval drugs. There was a little dip where the authors actually said it was probably due to the fact that both FDA and sponsors had to align on the new guidance and kind of make sure that they were following it correctly. But really, all in all, there's about 10% of drugs to date, orphan drugs to date, that have a PRO endpoint in their labeling. One of the other things I also wanted to take the opportunity here to touch upon is a little bit of the reason why this could be, and it is because we could talk about diseases as PRO-dependent diseases and then PRO-independent diseases. And so, for example, irritable bowel syndrome, asthma, pain, as we heard before, those would be PRO-dependent examples of diseases, while diseases that rely on a biomarker measure who need the assessment of a clinician, such as diseases of the circulatory system, or also diseases where the patients are not symptomatic, such as metabolic diseases. Those could be labeled as PRO-independent diseases. And obviously now I was talking about, all diseases in general, not just orphan diseases. So even though the voice of the patient is important, it cannot always necessarily be included in a clinical trial as an endpoint. I also want to emphasize that even when PRO measures are not primary or secondary endpoints, as Michelle said, they can be used as supportive information for an FDA submission, basically restating the benefit to the patient in addition to the measures that have been used that show statistical significance. Next slide, please. So in general, and I think this is probably more from the clinical development perspective, when we decide of how we select a PRO, I think there's a few different things that are very important. And PRO selection, just to state up front, should really happen early on in development, and I'll go over that in a couple of slides. But I think one of the items that is really important, as anybody would understand, is the mechanism of action of the drug. So if you follow on the horizontal on top, if you're expecting to be developing a disease-modifying treatment, you may want to look at changing of the disease course, and you may want to look at a multitude of symptoms that change over time. For example, inortersen, which is a drug that Ionis developed for the treatment of polyneuropathy of hereditary transthyretin-mediated amyloidosis in adults, one of the core primary endpoints was actually the Norfolk quality of life, diabetic neuropathy, which encompassed quite a few items. Now, if your mechanism of the drug focuses on one specific symptom, then obviously you would want to track that symptom and look at improvement in that symptom. So an example here would be suvorexant, a prescription medicine for insomnia for adults, and then the PRO that was used to support labeling focused on patient estimated sleep latency and patient estimated total time. Next slide, please. One of the other aspects that is important in PRO selection, obviously from a clinical development perspective, is the disease in the patient population. So again, following on the horizontal line, a common disease may already have defined disease stages, with clear symptom tracking. And so there may already be PRO available. And in this case, I mentioned a measure like irritable bowel syndrome. You can use Bristol stool scale or diary to record abdominal pain levels to really track that symptom. Now, for rare diseases, this is often different. Heterogeneous patient populations are involved in rare diseases. The disease progression is sometimes unknown or very early on in the knowledge base. And then the disease progression can also vary based on the age of onset, and so the age of patient and where they are in their disease. So if you are one of the first few companies that are trying to develop a medication for rare disease, it is highly unlikely that a disease-specific PRO already exists. So there's a few mitigation strategies, obviously, that you can use if you still want to obviously make sure that you address the patient's symptoms. You can see if there's existing PROs available that focus on specific symptoms. And in myotonic dystrophy, for example, they do have gastrointestinal issues. So you could say, well, maybe you can use one of the diaries to record abdominal pain or the Bristol stool scale. But then again, those symptoms might be different in myotonic dystrophy than in irritable bowel syndrome. So the tool might not cover everything. So in that case, one needs to answer the following question. Are there differences between young and old? And how can you go about to really find a PRO or develop a PRO on time for your clinical trial? Next slide, please. So as I said, to make the determination for the need, let's say, of a new disease-specific PRO, that should really happen early on in development. Best scenario would be actually during discovery phase, because I think we all know that it takes time to develop a solid validated PRO. You may also require to establish natural history studies. They may or may not be ongoing at the time. But I think that for diseases for which adequate measures don't exist, but for which the patient report is really necessary to assess treatment benefit, it is really reasonable to assume that the sponsor will invest time and resources and work with academic collaborators and these patient advocacy groups to really try and develop that really needed disease-specific PRO. And just quickly, because I will touch upon that later again, Approval is one thing, obviously, for a drug, but approval doesn't always mean access. And so we have to make sure that when we develop a medication, we keep in mind that we really need to get the medication to most of the patients. Next slide, please. Quickly, just here to mention that, you know, as you saw from Chad, how he developed the MD-High, I think obviously patient input was really important there to develop the PRO. But I think patient input can be done in numerous various ways. Participants are often invited for advisory committees, or at least they should be invited for advisory committees, to provide input on what is really important to them, and you know what they're expecting from a medication to benefit them. And I think, you know, all the different ways I'm listing here for patients to participate and engage. One of the ultimate purposes, I think, is that we need the patient's perspective and to understand the risk and the benefit. And the same obviously goes for the patient. I think ultimately, with the drug that we have for SMA, if you need an intrathecal injection every 4 months, you need to understand if the patients are willing to do this. But it will obviously depend on the amount of benefit that the patient gets out of that. So this might be different for a spinal muscular atrophy population versus an Alzheimer's or Huntington's disease population. Next slide, please. So I now just wanted to discuss myotonic dystrophy briefly. As you all know, this is a toxic gain of function RNA disease, part of the triplet repeat diseases. So CUG repeat are expanded in myotonic dystrophy, and they sequester a muscle blind, which then leads to alternative splicing pattern in all the cells. It's a rare autosomal dominant genetic disease. And unfortunately, it has this phenomenon of anticipation where every generation has the disease earlier and more severely than the previous generation. And so it's quite devastating because normally when a child gets diagnosed, you then also diagnose the mother and one of the grandparents at the same time because they had the disease in a milder form. It has a broad spectrum of symptoms, including muscle function, GI tracts, cognitive issues, as I said before, because of anticipation, you have a congenital form, a juvenile, and then an adult. Next slide, please. So when we first started to work on myotonic dystrophy, obviously one needs to determine, are you trying to work on a disease-modifying therapy or symptomatic therapy? With our RNA-based approach, we actually were able to try and get to a disease-modifying therapy. And what we first did is we knew we had to address systemic and CNS symptoms, but we also knew that our antisense oligonucleotides do not cross the blood-brain barrier. So we first focused on systemic issues because we could measure muscle strength and myotonia. And so it was a bit easier to track symptoms there. But I think it is always important to understand what's bothersome to the patients. And over time, over the last few years, while we know that muscle weakness and myotonia are considered prominent symptoms of the disease, a lot of the patients actually say that their CNS symptoms are more bothersome, because those include excessive daytime sleepiness, fatigue, and cognitive dysfunction. Next slide, please. We are very lucky that Chad had just finished developing the MD-High when we started our clinical program. And as he mentioned, this is a scale that include different subscales. And as you can see, there's different questions per the different subscales. We were lucky that there was already natural history studies that had been established for this disease, and the MD-High had been tested. And so it was easy for us to include this in our trial. And so we included this in our multiple ascending dose study in patients with myotonic dystrophy 1. I will unfortunately not show results of this here because we did not reach a high enough pharmacokinetic profile in our patients. And so unfortunately I have to go back to the drawing board for this disease. But what I think is really important is that the breadth of the MDI would have allowed us to actually better understand which domains were affected by the drug. Sure, we said we wanted to affect it or to develop a disease-modifying drug, but how sure would we really be of that? And so I think items here that would have been really interesting for us to see if we could have reached changes were gastrointestinal issues, following issues, to really see if the drug would get into those tissues. An additional benefit of a scale like this, for example, is that in clinical development, we all struggle with trying to assess as many scales and outcome measures as we can in the trial, while at the same time not overburdening the patient. There's no use to do 4 different muscle tests in a row because, you know, on the 5th muscle test, they cannot, you know, give their best anymore because the patients are pooped out. And so again, this would have allowed us to better understand, are there GI improvements? Are there swallowing improvements? And if that had turned out to really be one of the items that was the most important, then you could follow up in the next trial with maybe more specific tools or things like that. Next slide, please. In addition to well-developed PROs such as the MD-High, I think what's important to obtain is also additional qualitative information from the patient. As drug developers, we learn of, you know, disease we work on in multiple ways. Sometimes we have maybe had the chance to treat patients with a disease that we are working on, but oftentimes we have not, especially not in the rare disease field. So it's important to truly understand the patient from the disease from the patient and caregiver perspective. And so for myotonic dystrophy type 1, we were lucky that in September of 2016, Myotonic Dystrophy Foundation set up the first externally led patient-focused drug development meeting. And so that really provided a lot of information on what the patients